So today we're going to talk about organization of the nervous system, the first of three or four lectures on this. Get this down to a lower. And there are four fundamental dichotomies that you have to understand. And they have all been touched upon, some explained in their entirety by Dr. Larson, but I will quickly review them again. We'll be concentrating on the fourth one, at least by more than the others. One is the distinction between the central nervous system and the peripheral nervous system. So, woman there in the white glasses and the maroon T-shirt. This is not a hard question. And that always makes people feel guilty if they don't know the answer, right? What is the distinction between the central nervous system and the peripheral nervous system?
Central nervous system includes the brain and the spinal cord, everything between. Peripheral are all the spinal nerves coming out.
And that's an excellent and perfect answer. The central nervous system is brain and spinal cord, and the peripheral nervous system is everything else that's part of the nervous system. Afferent versus efferent. This is one that Dr. Larson touched on, and I'm going to go into a bit more detail. These words come from the Latin word ferre, spelled with two R's, which means to lead. A is short for AD, which means to, and E is short for EX, which means from. The afferent part of the peripheral nervous system, they apply to the peripheral nervous system, really, or is the part which leads to the central nervous system. The efferent part of the peripheral nervous system is the part which takes information away from the central nervous system and distributes it out to the body. Now, I've just defined these in sort of functional terms, but there are genuine anatomic correlates. There are nerves, not many, but there are some nerves which are purely composed of afferent material, that is axons or cells that lead from the periphery to the central nervous system, and others which are purely concerned with efferent things, taking information and sending it out of the central nervous system to the periphery. And there are some parts of the central nervous system which are concerned solely with processing information brought to it, therefore solely afferent processing, and some parts of the central nervous system concerned solely with sending stuff out. They're therefore efferent parts of the central nervous system. So here is a drawing, my drawing, of a cross-section of the spinal cord. We don't do much, as Dr. Larson said, we don't do much by way of central nervous system anatomy, but I would think people should have an understanding of the basic, the most rudimentary anatomy of the spinal cord. So this is a cross-section. Posterior is up at the top, and you can see where the anterior is. And when you look at a cross-section of a spinal cord, you see some things that get names. There is a hole down through the middle of the spinal cord which contains cerebrospinal fluid that is called the central canal of the spinal cord, and it is connected with bigger holes inside the brain which are called the ventricles of the brain that carry cerebrospinal fluid. There is a septum, sort of a connective tissue septum that runs down the middle of the dorsal side or posterior side, which is called the dorsal median septum. Its name tells you everything you need to know about it. And then there is a groove in the ventral side called the ventral median fissure. Again, a fissure is the word meaning groove, so it tells you everything you need to know about it. And the spinal cord looks like this. Its entire length gets bigger and smaller in some places, but pretty much this is the way it is. There's also a zone in cross-section, always looked to me like a butterfly, which is called the gray matter, and that's where all of the cell bodies of the neurons are located. They are not, the cell bodies have no myelin at all. Myelin, you may know from your undergraduate career, is a lipid substance which surrounds axons and promotes the speed of transmission down them. And since there are very few, only the beginnings and ends of axons here, and it's mainly cell bodies, it's not very myelinated, so in a fresh specimen, if you take a spinal cord out of a dead person and cut it, this looks gray because myelin is fatty and looks white. And all this other stuff, which has all of these highly myelinated axons running up and down the spinal cord, some from the brain, some to the brain, some to different levels of the spinal cord, that myelin makes this appear white in a fresh specimen. The gray matter has different regions. Up near the top, near the dorsal part, it's called the dorsal horn of the gray. Down near the bottom part, near the ventral part, it's the ventral horn of the gray matter. And in certain regions, not all, but in the middle of the cord, there is this little bump in the gray matter there, which is called the intermediolateral column, or lateral horn. You probably wish that I would call it lateral horn, but its proper name is intermediolateral column, and that's pretty much what I'll call it all the time. It turns out that the dorsal horn of the spinal gray is, in fact, afferent. It is concerned only with processing information brought from the periphery to the spinal cord. Those cells process afferent information. The ventral horn and the intermediolateral column, it's essentially the ventral half of the gray matter, is a efferent gray. The cells there are only involved, really, in sending information out of the spinal cord to the periphery. Now, this is what Dr. Larson talked about. It turns out that most afferent information brought to the spinal cord is sensory information. And by sensory information, we mean able to reach consciousness. Information that you can be aware of. But you know, not all of the information, as she mentioned, that's brought into the central nervous system can come to consciousness. For example, if I were to ask you now, is your blood pressure high or low, you would have no idea. And yet the central nervous system gets tons of information about blood pressure. If I were to ask you, what is the acidity of your stomach, you would have no idea. But this kind of information, that, oxygen concentration in blood and things that I can't even remember, is coming into the nerves. Yes, sir?
Does the color relate to anything, like all the afferent nerves are in the posterior part of the spinal cord, or you just did it for color?
Every cell body, remember the gray matter is full of cell bodies. Every cell body here is going to receive afferent. It's going to have an axon coming from the periphery, synapsing on it, and is going to be involved in the processing of that sort of information brought to the spinal cord. Every cell body here is going to send an axon out to the periphery. All right, so because most of the afferent stuff is sensory, and only some part of it that we've discussed here, blood pressure, oxygen concentration, your stomach acidity, and those things are non-sensory afferent. It means you have no way of having a conscious awareness of those things. We are pretty much in this course only be concerned with sensory, the sensory component of the afferent system. All right? Similarly, efferent is largely motor. By motor, I mean going to the only two tissues of the body that can actually do anything for you, which are muscles and glands. Your ability to have an impact on the world at large is due to the muscles and glands that you have. And those are the motor tissues of the body, muscles and glands. And any axon or cell that sends an axon out to a gland or a muscle is part of the motor system. That's a subset of the efferent system, because there are other cells in here that go and influence bone metabolism. When we get to the brain, there are cells that influence the way your ear functions and the way your eye functions. Those are all efferent things. They go out of the central nervous system to influence the functioning of other organ systems, but they don't go to glands or muscle, and therefore they're not motor. We are concerned in this course with the motor portion of the efferent system and the sensory portion of the afferent system, and those are the dominant parts anyway. And so we can call this, for our purposes, the dorsal horn is the sensory gray matter of the spinal cord, and the intermediolateral column and ventral horn is the motor gray matter of the spinal cord. And ignore non-sensory afferent and non-motor efferent. Now we get to, again, very important, distinction that Dr. Mark Larson mentioned, visceral versus somatic. This is best defined, in my opinion, by looking at the motor tissues of the body. Now, what are the motor tissues of the body? I haven't told you this 30 seconds ago. What are the motor tissues of the body? Muscles and glands. That's right. That's all. No another. If there is, I'm not sure what the next slide says. Maybe I should. Yeah. There are two kinds of these motor tissues. I don't mean muscles and glands. I mean, that's two kinds too. But I mean, within that group, muscles and glands, there are two kinds of motor tissues. One are somatic motor tissues. That's the striated muscle. The muscle you would call voluntary muscle, but which is properly called striated muscle. All right? You have voluntary control over most of your striated muscles, not all, and sometimes they are involuntary actions, but that is the one somatic motor tissue of the body. All of the other motor tissues are visceral motor tissues. So, what are the visceral motor tissues of the body? No, the non-striated muscle motor tissue. Smooth muscle is one. Cardiac muscle is the second. Gland is the third. That's it. Just three of them. Smooth muscle, cardiac, and gland. Those are the visceral motor tissues of the body. All right, so now we really clearly define this by terms of motor tissue. Somatic motor tissue, striated muscle, visceral motor tissue, cardiac muscle, smooth muscle, gland. The visceral, there are different parts of the nervous system concerned with controlling these two different types of motor tissues. The visceral motor system, the part that's concerned with innervating visceral motor tissues, is called the autonomic nervous system. That's what it is. That's a synonym. Visceral motor system, synonym autonomic nervous system. There are two kinds of bodily structures in the world. All right? There are visceral structures and somatic structures. Now here, it's easier to define a visceral structure than there is a somatic, was just striated muscle, and all the other motor things were visceral. A visceral structure is a structure composed primarily of a visceral motor tissue. So a visceral structure is composed primarily of cardiac muscle, or primarily of smooth muscle, or primarily of glandular tissue. Sometimes you can combine them, like the gut, which has lots of glandular tissue and lots of smooth muscle in it. But arteries and veins have smooth muscle in their walls, no glands really. They are visceral tissue. The heart is obviously a visceral structure. All right? Everything else is a somatic structure. Unless you are composed primarily of smooth muscle, cardiac muscle, or gland, you are a somatic structure. Now, the distinction is important, not just generally, but because, again, within the central nervous system. Oh, actually, I have to do one other thing that occurs to me, as I knew the slide would tell me. We know that the visceral motor system is equal to the autonomic nervous system. That is different from the somatic motor system, which just goes to striated muscle. If you can identify a structure as a visceral structure, any afferent information that comes back from it is called visceral afferent information. If you can identify it as a somatic structure, any afferent information that comes from it is somatic afferent information. And they differ quite dramatically. Number one, all somatic afferent information is sensory. Every sort of information that is sent to the central nervous system about a somatic structure is capable of reaching consciousness. And that is not the case with visceral afferent information. Much of it, we've discussed them, is incapable of reaching consciousness. So all somatic is sensory, but not all visceral is sensory. It depends upon the structure that we have defined. Now, let's then, since I told you in this course, we're not really concerned with non-sensory afferent very much. Now let's limit our consideration to the differences between somatic sensory information and visceral sensory information. That is, the actual stuff coming back from either a somatic structure or a visceral structure capable of reaching consciousness. And there are considerable differences here. Number one, yes.
What about proprioception?
Proprioception is capable of reaching consciousness. That is why if I ask you, is your elbow flexed, you will say yes. And if I ask you, is your elbow straight, you will say yes. Now, there may be, and you can tell if a muscle is contracting a lot or not contracting a lot, because most proprioception is capable of reaching consciousness. Now, the differences. In terms of the sensation that comes back from a somatic or a visceral structure, quality is different. I don't know if I have this on value. I'll tell it to you because I'm not sure if it's on the slide. Any sensation that comes from all visceral sensation is unpleasant or painful. It may start out as just unpleasant and become painful, such as when you have eaten too much food, if you sense your stomach getting full and you feel a little discomfort, if you're stupid enough to continue eating, it'll eventually get so full as to hurt. All right? I'm trying to think of another example of this. I guess actually when your rectum is full too. Your bladder. Let's take your bladder. But, initially your bladder, it fills up at the end and you're squirming around, you're uncomfortable, and after that it starts to really become painful. So it can start out as uncomfortable and become painful, or it can be painful from the get-go. You have a heart attack, there's no such thing as, oh, gee, my heart's uncomfortable now, and in a couple of days it's painful right away. Same with gallbladder, inflammatory. It's always painful from the start. Visceral sensation is always uncomfortable and or painful. Somatic sensation, which you are clearly aware of, may be uncomfortable and painful. People can burn you, can cut you, that's uncomfortable and painful. Or they can tickle you and do other things that make you pleasantly surprised. So somatic sensation can be either painful or pleasant. Visceral is always bad. Or not bad, you just don't like it. Localizability. You know precisely where you have been touched, cut, or burnt on your skin. If you pull a muscle, pretty much you can point to where the pain is in that muscle precisely. Visceral sensation is very poorly localized. You just have a vague sense, in my abdomen something hurts. You're having a heart attack, an elephant stepping on my chest, but I don't, but it may be only like the tiniest little bit of your heart muscle which is causing the source of the pain, but you cannot say, oh, it's a bit of the heart, 2 centimeters to the left and 3 centimeters down. It's just a pain that's very diffuse. So visceral sensation is poorly localized. Somatic sensation is well localized. Stimuli that elicit pain now. Now, we already know somatic sensation doesn't have to be painful. So now let's just look at pain itself. The stimuli, there are 7 stimuli that elicit pain. These are listed here. Let's look at somatic pain. You can think of an instance in yourself of every single time where you cut yourself, it hurts, where you burnt yourself, or you're too cold and it hurt you. Someone pinches you real hard, that hurts. Someone grabs, your cheeks and goes like that, that hurts. Cramping, you have a muscle cramp like in your biceps or your gastrocnemius or something, that hurts. Obviously, inflammation of the skin, you have an abscess or a pimple or something, that can hurt. And ischemia, one you're less sensitive to because it doesn't happen so much in people your age, is that, and that means inadequate blood supply. That is, you're not bringing enough oxygen to a region of tissue. That is also a painful thing. So these are the 7 things that can cause pain in a somatic structure. Only these 4 are capable of eliciting pain in a visceral structure. Take a look at your bowel. Well, if you go in a living person let's put it this way. Let's first look at the first 3 that don't cause pain in a visceral structure. If you can get through the skin into your abdomen without screaming in horrible agony, as some crazy people can do, actually, you can actually do a lot to your gut without any pain emerging. You can cut it, you can burn it, You can crush it in a thing, and it will elicit no pain at all. So if these crazy people who do operations on themselves, they only have to get through the skin, really, and the muscles. Once they're in there, they can muck about in there all they want, and it really doesn't hurt, unless they were to stretch the tissues attached to the gut. That will hurt. If they cramping, everybody's had an abdominal cramp. Your intestines get real tight, and that is a painful thing, a sustained muscle contraction. Obviously, inflammation, gastritis, any kind of inflammatory condition of the bowel, cystitis, an inflammatory condition of your bladder is a painful condition, and ischemia, which is one of the most common causes of pain in the visceral structure. That's what a heart attack is. It's ischemia of the heart. But these are the four things which cause both visceral and somatic structures to have pain, and three of them don't. The visceral structures just don't respond to them. Now, we talked about there being an anatomical correlate between sensory and motor. There being some things which are only sensory, some which are only motor. There are some things within the sensory portion which are only concerned with processing somatic sensation. Those are the red bars here. The cells in those red bars are only concerned with getting input from somatic sensory neurons and processing somatic sensory information, whereas there's laminae of cells in the dorsal horn which get input from both somatic and visceral structures and get both of those kinds of information. Those are called viscerosomatic. So there's a viscerosomatic sensory gray, cells that get inputs from both somatic and visceral structures, and the inputs they get from somatic are generally painful. And then the somatic, which doesn't have to be painful, portion of the visceral gray, of the sensory gray, sorry.
Is the striation
What?
Is the striation specific to the dorsal side, or is that just
The striation is specific to the dorsal cells.
Yes.
The dorsal horn cells are in laminae. And the laminae, and there's numbers and names for them, and maybe you'll talk about it in neuroanatomy, maybe you won't. But that lamination is specific for the dorsal or sensory gray of the spinal cord. In the motor gray of the spinal cord, there's also differentiation between visceral and somatic, but here it's not lamination. Here you've got everything in blue, more or less, being somatic motor, meaning these cells are concerned only with the innervation of striated muscle, the cells in that blue area, whereas the yellow is the visceral motor gray. And the cells in that yellow area, that intermediolateral column, are concerned only with innervation of cardiac smooth muscle or glands. So there is a distinction anatomically as well as the functional one that we made up. And now we get to what the rest of this lecture is really going to be about, which is the distinction between the body wall and the body cavity. And I'm not going to reiterate what Dr. Larson said, but I want to say this about it. The body wall is overwhelmingly made up of somatic structures. There are some smooth muscles. What are the smooth muscles in the body wall? Oh, gosh. Who answered that? All right, so now you have to answer them all. Our blood vessels, yes, blood vessels are visceral structures in the body wall because they are largely smooth muscle with a lining inside and a lining outside.
Erector pili muscles?
The erector pili muscles, which are smooth muscles which cause you to get goosebumps. Sebaceous glands probably don't get innervated, but they are a visceral structure too. But there's another gland that does get innervated. Sweat glands. So those are the visceral structures in the body wall. But those don't really make up very much of the body wall. The body wall is mainly skin, muscle, tendon, fascia, bone. And those are all somatic structures. And therefore the body wall is overwhelmingly made up of somatic structures. Now, whoever or whatever it is that has created vertebrates on Earth created spinal nerves for the purpose of innervating the body wall. That's why we have spinal nerves, to innervate our body walls. That means, since the body wall is overwhelmingly made up of somatic structures, spinal nerves are overwhelmingly made up of somatic sensory and somatic motor axons, because those are the things you need most of to innervate the body wall. So now, and the pattern by which the body wall is innervated is a segmental pattern that is created in embryogenesis in a way that I hope everyone now knows about, since you should have gone through at least the first 2 lectures. You haven't till midnight tonight to do the first embryo quiz. If you got as far as folding of the embryo, which is a little bit into the group 2 lectures, you will know even more about what I'm going to talk about, not necessarily. This is an actual photograph, or a photograph of an actual 4-week human embryo. Not nearly as attractive as my representation of a 4-week human embryo. Mine has been cut in, I would love to say this is a median sagittal section, because it was, sort of like that. But in fact, I've tried to give some 3-dimensionality here to some of these structures, and so I'll talk. I mean, the gut is cut in median sagittal section of my 4-week folded embryo. The heart, however, I tried to give a sense of it being a tube, a heart tube at this point, and I tried to give a sense of the aorta being this big artery along the back of the gut. Again, I tried to give some 3-dimensionality to the neural tube in green, and of course, the somites sit, if this were a true median sagittal section, the somites would sit in front of the plane of the blackboard, the left-handed somites, and there'd be the right-handed somites and they're behind the plane of the blackboard. So this is not really a section. It's a schematic drawing of a 4-week embryo. If we take off the top of that 4-week embryo, take off the ectoderm from the back of the 4-week embryo, and we look down onto its surface, this is what we will see. Now, these somites are, as you know from your embryology, the fundamental structure that cause segmentation of your body. They create segments from what was a non-segmented structure. Here is our neural tube that we would see looking down onto the back after the ectoderm is taken off. That neural tube has an expansion at one end, which would become the brain, and a much longer. Well, not at this stage, it's not much longer, but it will be when you're an adult. A much longer thing called that will not expand so much, which is the spinal cord. Here are all of the somites that are caudal to the head region. There are some somites and some other things called semitomeres, which are on the opposite side of the brain, are very similar to somites in many ways. You'll learn about it when you do head embryology. But these are the somites of what we would call the neck and trunk region of the embryo. And they are numbered here. I can never remember how many there are, but the slide tells me that there are 31 complete somites on the left, 31 complete somites on the right. And then down near the tail end of our embryo, there are 3 sort of degenerating or degenerative or abortive somites. There used to be even more. I mean, we used to have a longer tail in embryogenesis, and we lose a lot of the somites that were created back here, but we do still retain 4 abortive ones. So there's 31 complete, and I don't really care about the others. If we do a cross-section through our 4-week folded embryo, right through the neural tube and the somite, and of course we'll see the aorta will be there, and the gut will be there, and then some other things we don't know about yet. So here's the cross-section in this picture. It's going to look like this. Here's the neural tube. Here is the aorta. I didn't draw in that other section. I don't know why. I don't know. I was too lazy. A structure you know about from your embryology, the notochord, coming out from Henson's node, inducing the folds which form the neural tube. That is always directly ventral to the neural tube. And here is the gut. I don't think but you saw it in the previous slide. I didn't label it. You also know from your embryology that as the neural tube is formed, on either side of it, the crests of the neural folds separate off and become this tissue called neural crest tissue. At first, a column of neural crest runs the whole length of the embryo on the left side, A column of neural crest runs the whole length of the embryo on the right side, but when the paraxial mesoderm segments into somites, the neural crest segments along with it. So you get, instead of a column of neural crest, you get a clump of neural crest cells for every given somite on the left, and a clump of neural crest cells for every given somite on the right. We're going to talk now about somites and their contribution to the body wall and how this relates to spinal nerves, whose job it is to innervate the body wall. First thing that somites do is they split into a ventromedial portion and a dorsolateral portion. The ventromedial portion of each somite is called a sclerotome. Sclera means hard and tome means cut. So I don't know why it's called, it could be called sclera something. You'll see why the tome doesn't make a lot of sense to me. Yeah, because it's a cut part of it. I don't know. Yeah, sclerotome. That's what the dorsomedial part of each somite is called. The ventromedial part. The dorsolateral part of each somite is called a dermomyotome. You would deduce from its name, from this name you would deduce it becomes something hard. Otherwise you wouldn't call it sclerotome. From this name you would deduce it becomes skin and muscle. And to a certain extent it does. I may mention it in detail later. Well, to a large extent it does. But I will definitely mention it later, because I've just left you puzzled now. Let us follow the sclerotome part first. Here is that looking down view, 31 complete somites on each side, and we've now divided each somite into a dermomyotome and a sclerotome. So we have 31 dermomyotomes on the left, 31 on the right, 31, and actually these abortive ones only become sclerotomal. So we actually have 34 sclerotomes on the left and 34 sclerotomes on the right. Now, the next thing is that each sclerotome divides. And it divides into a cranial half and a caudal half. If we make a cut here, a transverse section here, you will see no sign of that having happened. All right? The only way you could have told that that happens if I made a cut right down in the middle between the two halves, which would be hard to do. But, so this picture doesn't change. But I labeled it as cranial half because I know that that's the section I went through. So each sclerotome divides into a cranial half and a caudal half. And then the sclerotome cells, both cranial and caudal halves, start to migrate medially towards the neural tube and notochord. But their migration is not simple. The cranial half of a sclerotome migrates medially and cranially, and the caudal half of a sclerotome migrates medially and caudally. And after this migration is complete, you can see here we've got the caudal half of sclerotome 1 actually being closer to the cranial half of sclerotome 2 than it is to its own cranial half. And you know, I can't remember the numbers. This is a sclerotome 11. The caudal half of sclerotome 11 is now closer to the cranial half of sclerotome 12 than it is to its own cranial half. And then these sclerotomes, having migrated this way, fuse with one another, the cranial half of one fusing with the caudal half of another, to create something which is no longer segmental in nature. The basic segmentation was laid down by somites. The basic segmentation is maintained by dermomyotomes, which have not undergone this bizarre splitting and migration. But what has been produced by the fusion of a caudal half of a sclerotome with the cranial half of the one behind it is a structure which is intersegmental. It lies between segments. These fused sclerotome halves are the future vertebrae of your body, and vertebrae are intersegmental structures. I'm showing you here a sclerotome cells have migrated surrounding the neural tube, surrounding the notochord, saying to themselves, I want to become a vertebra now. I'm going to ask, does anybody know from either what I may have said in some embryology lecture or from some other source, what happens to the notochord as it becomes surrounded by these sclerotome cells?
How's the intervertebral
dissolves is your one answer. Someone else may have said a different phrase of the same meaning. It gets destroyed by sclerotome cells, except in one location. Vertebrae, in between vertebrae are these spaces called intervertebral spaces. And intervertebral space will develop into an intervertebral disc, binding the two bodies of the vertebrae together. The notochord persists as the nucleus pulposus of the intervertebral discs. So it does persist in that way as the nuclei pulposi of the intervertebral disc, but everywhere else, the vertebrae, as they form, destroy it. Now here is the vertebra formed by the cells from the cranial half of one sclerotome and the caudal half of the adjacent sclerotome. And I'm just labeling the parts of the vertebrae here for you. You won't read about this in theory. If you delay all your reading till after the relevant section, you won't have read about this. But vertebrae have different regions to them. This would be the lamina of the vertebra. This ridge here, or bar there, would be the pedicle of the vertebra. From the junction of the lamina and pedicle comes a little bump called the transverse process. From the end of the pedicle, or the medial end of the pedicle, comes a process called the costal process, which is related to rib formation. And then this thing, which you hoped I would have labeled the body, but didn't, I labeled the centrum, because in fact, in embryogenesis, the vertebra forms of these two things, a neural arch and this thing called a centrum, and later these will join together to form a body, but the body is actually, of an adult vertebra, is actually comprised of the centrum plus a bit here of what we might call the pedicle of the neural arch. So the body is more of a vertebra, is a larger or more all-encompassing structure than just the centrum. So, but there's a reason I gave that name there. I wanted you to know that. You'll see in a second.
Dr. Stearns?
Yes.
When is this all happening? What week?
This is all, I don't have any idea. Honestly, I can make a wild guess. Let's put it this way. I can tell you that by 8 weeks, the little creature inside the mommy looks just like a human being, so that's why it changes its name to fetus. So it's well before 8 weeks, all right? But, and probably doesn't begin really to get going until after folding, so I'm guessing somewhere in the second month of life. But if anyone knows different, I'll buy it. Yeah.
When the dermal myotome are, migrating to form the vertebra,
The sclerotome.
right, the sclerotome,
Yeah.
are migrating to form the vertebra, how do they form the circle? What's the 3D? I'm getting confused on the 3D version of this.
Well, they migrate medially, and then the cells just spread around.
Okay.
They spread around the entire neural tube and notochord. No?
Mm-hmm. That makes sense.
They just spread. I mean, these cells are not attached to one another. They leave as individual cells from the sclerotome, spread out to envelop the neural tube and notochord. And I don't know how else to say it, to be honest with you. So now I've drawn in the vertebrae. I've just taken what before were pink, and I said was a joined caudal and cranial half of the sclerotome, and just turned it into yellow, indicating that it turned into bone, and we guess when that might happen. Well, let me first get 100% straight. It does not turn into bone until much later in fetal life. First, it's going to turn into cartilage, and you'll get a cartilaginous vertebra. Then that cartilage will turn into bone. And so when I said it looks like a human at 8 weeks, I meant the vertebrae were cartilaginous by 8 weeks. Then over the next few months, that cartilage will turn largely into bone. No, but the centrum actually does not fuse with the neural arch until after you're born. So it's not even a complete bony structure when you're born. Timing, I don't care about timing for a lot of these things. Some things are important, some things aren't. All right, so now, but I've said, okay, it's bone, it's a vertebra. But in an embryo, in a fetus, it's not a bone. All right. The costal processes of some of these in the middle grow very big. They grow very large. The costal processes first as migrating sclerotome cells and then turning into cartilage. And those will become actual ribs that we will see in the adult. The costal processes of the other vertebrae don't become large. They haven't disappeared. Every vertebrate, and we'll talk about this in a week, every vertebrate still has something that's homologous to a rib. It just is a small rib. It doesn't look like a rib. But some of them have real long costal processes and become real, genuine ribs. And the ones that do are called thoracic vertebrae. And the ones in front of them, the 7 that don't have long costal processes, in front of the thoracic vertebrae are called cervical vertebrae. And all the rest of the ones behind the last rib-bearing vertebra are not called thoracic. Now 5 of those, there's 5 which do not fuse with one another, and then there's 5 which do fuse with one another. The 5 which fuse with one another are called sacral vertebrae. And this is what a sacrum looks like. So it actually has some holes in it, which I didn't show in this picture here. It has to have holes because the nerves have to come out somewhere from, because the vertebrae envelop the neural tube, and the neural tube is going to give rise to nerves. So there have to be holes for the nerves to come out of. So sacrum has to have holes. So I just drew little splits here to indicate to you that these 5 vertebrae that fuse into the sacrum do have holes at certain locations for nerves to get out. The 5 vertebrae between the fused ones and the last rib-bearing vertebra are called lumbar vertebrae. And the 4 vertebrae caudal to the last of the fused sacral vertebrae are called coccygeal vertebrae. You can count up how many there are if you want to add all these numbers together. I don't know. All right. Here are all of our vertebrae: cervical, thoracic, lumbar, sacral, coccygeal, and the dermamyotome. Now, I don't mean to suggest, certainly I don't mean to suggest that all of this is happening to completion and the dermamyotomes are just sat around waiting for this all to complete. Everything, in embryogenesis, everything happens simultaneously. But I can't discuss it as if it was all happening simultaneously. It's hard enough to understand it if you discuss it sequentially. But so let's pretend that all did happen and it's all completed, and now we go and look at the dermamyotomes, even though we know it's all happening simultaneously. There are, after this has happened, 8 dermamyotomes sitting cranial to the first rib-bearing vertebra. So if the first rib-bearing vertebra is the first thoracic vertebra, and that's the first rib, and that marks when the chest starts, when the thorax starts, then you've got 8 dermamyotomes sitting cranial to the chest, which are in the neck. So you have 8 cervical dermamyotomes. Now, this is obviously the first dermamyotome in the chest, so that's T1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. If I were in charge, if I had been 400 years old, and I would not have called that the last of the thoracic dermamyotomes, because it's below the chest. But whoever named these things said, no, we're going to say that that is the last of the thoracic dermamyotomes. Even though it's not in the chest, it's below the chest. It's more closely related. And therefore, if that's the last of the thoracic, you have 1, 2, 3, 4, 5 lumbar, 1, 2, 3, 4, 5 sacral, and there's only 1 coccygeal. And what this means is you've got, everything is screwy number-wise. Because the first cervical dermamyotome actually lies cranial to the first cervical vertebra. And the second cervical dermamyotome lies cranial to the second cervical vertebra. And the 8th cranial dermamyotome lies cranial to the first thoracic vertebra. But after that, all the dermamyotomes lie caudal to the vertebra of the same end. That's just the way it is. You can't change the nomenclature this many hundreds of years after everybody agreed on it. So you've got a situation where every dermamyotome has the same name and number as the vertebra caudal to it, except C8, because which has its own name and number independent of anything. There are only 7 cervical vertebrae. There are 8 cervical dermamyotomes. After that, every dermamyotome does have the same name and number as the vertebra cranial to it. You have 8 body segments in the neck. 12 body segments that are said to be in the chest, 5 body segments in the lumbar region, 5 in the sacral, 1 in the coccygeal. We'll come back to what happens to all of that in a moment. The neural crest clump, for every dermamyotome, there is a neural crest clump. It starts to migrate ventrally on both sides, each clump. As it does so, each clump divides into two. The two separate from one another, with one of the subclumps taking up a position between the pedicels of adjacent vertebrae, and the other subclump taking up a position really pretty much adjacent to the beginnings of the costal process of that vertebra. The clumps which take up a position between the pedicles of adjacent vertebrae are destined to become the dorsal root ganglion or the spinal ganglia. The clumps which take up a position near the beginnings of the costal processes are called paravertebral ganglia because they're alongside the vertebrae, and we will talk about them on Friday. Now I'm going to concern myself with the subclumps that are between the pedicels of adjacent vertebrae, destined to become the spinal ganglia. All right. Next, yes.
Question.
You have to yell louder because I'm
Sorry,
old and deaf.
I'm wondering, is there a capillary network at this time?
Yes, these things, well, I can't tell you. Let's put it this way. These cells need oxygen and nutrients. There's a circulation established, as we know, at the end of the third week. Beginning of the third week?
No.
When?
At the end of the third week.
He says end of the third week, so he read your lecture notes more than you.
Yeah, there's a heart developing.
I think it begins at the end of the third week you get circulation. Until then everything is diffused in. Circulation begins at the end of the third week, and I'm guessing there are probably capillaries developed by this time. But you really have to keep these things alive. They're rapidly metabolizing. All right. So again, everything's happening simultaneously. We took the vertebrae, and then we took the neural crest migration. Now the dermamyotomes, each one divides into two. A dorsomedial portion of a dermamyotome, that's called the apaxial portion of the dermamyotome, means above or the axis. And a ventrolateral portion of the dermamyotome called the hypaxial portion of the dermamyotome.
Dr. Stanford.
Yes.
I have another question. Let me get terminology. The neural crest that moves in front of the developing rib.
Nearby, right?
Nearby. Okay. So, there's only developing ribs in the thoracic vertebra.
No, the costal process. I should have said, you're right to correct me if I said rib. Next to the beginning or the medial part of the costal process of every vertebra.
Oh, okay.
Every vertebra does have a costal process, and there's a ganglion that takes up a position very near the root of the costal process of that vertebra.
Okay, thank you.
Whether it becomes a rib or not. All right, so now we have every dermamyotome on left and right dividing into a dorsomedial apaxial and a ventrolateral hypaxial portion. I've shown that division here. So initially we had 31 somites, complete somites total. Then they became 31 dermamyotomes on either side, and now we have 31 apaxial portions on the left and on the right, and 31 hypaxial portions on the left and on the right. And I'm going to take a blow-up view of this region then of the neural tube, except you're going to see it doesn't look at all like this, and of these dermamyotomes, because I'm going to allow the neural tube to become a spinal cord, just for ease. So really what I've got here is a completely developed neural tube in the spinal region. I do have an apaxial dermamyotome and a hypaxial dermamyotome in that clump of cells in between the pedicles of vertebra from the neural crest. All right? Every single striated muscle of your trunk, of neck, everything below the head, the neck and the trunk, and of your limbs as well, which are outgrowths of your trunk. Every single striated muscle is derived from a dermamyotome. It turns out that, and this is a detail that I want you to forget immediately, the hypaxial dermamyotomes only become striated muscle. Apaxials actually become skeletal muscle and contribute to the dermis of your back. Now, that's a detail I don't think anyone has to know. That's why it's called dermamyotome. It used to be thought that both of them contribute to skin, but recent studies show that only the apexial contribute to skin. So, but the thing I do want you to know, because I don't care about embryology of the skin. It's a funny story, but it's about a guy long dead, so I wouldn't even get anything. But at any rate, the one thing you do have to know is all of the striated muscles from your head down are derived from these dermamyotome cells. Forgetting that little thing about the skin, that's what dermamyotome cells do, and they don't do anything else. I told you about the skin. Let's say that's what they do. They become striated muscles, and all the striated muscles come from them. That is their job, to become striated muscles. The cells that are going to innervate striated muscles are, by definition, somatic motor. Because striated muscle tissue is a somatic motor tissue. The somatic motor region of the spinal cord gray is in the ventral horn. Therefore, the cells in the ventral horn have as their job to innervate the muscles that will come from these dermamyotomes. So there are a whole bunch of cells in the developing ventral horn of the spinal gray whose job it is to innervate striated muscles derived from these things. So I'm going to say, and this is really what happens. These cells send out an axon. It's being drawn to the right place by signaling in the intercellular spaces or some miraculous region. An axon grows out. I don't know where this axon is going. You know where this axon is going? You do if you look ahead on the slides. Let's see where this axon is going. It's got to head to one of these dermamyotome things because that's what's going to innervate the muscles derived from it. That's going to a hypaxial dermamyotome muscle cell, one muscle that will come from that hypaxial. Here's another cell. It's sending its axon out. It will be drawn to an epaxial, all right? And hundreds of these cells will send axons out. Some will go to hypaxial muscles, muscles derived from the hypaxial dermamyotome. Some will go to muscles derived from the epaxial. Now, I'm only showing one each, but hundreds or thousands, maybe even, grow out to do this.
Professor, the axons sent out to the dermamyotomes or to the muscles once they've developed?
The axon, because all of this, everything is going simultaneously. These cells, these dermamyotome cells, are starting to differentiate into muscles. And as they start that differentiation, the axons are growing towards them. Right? It's not that the muscles get completely developed and then the nerves go out. Everything is happening simultaneously. So here are just these, I just picked the two cell bodies in the ventral horn, and one of them sending an axon out to the epaxial, and one of them sending an axon out to cells of the hypaxial. And I've shown now a little bit the hint that the epaxial dermamyotome cells are going to start to migrate onto the back or the dorsal surface of a vertebra. The hypaxial ones are going to start to migrate down into the lateral body wall of our embryo. So that migration is going on also, everything's simultaneous. Now, let's take a look at these cells in that neural crest clump associated with these dermamyotomes. These are sensory cells. A cell will, in development, send out an axon. That axon will split into two directions. Send one process towards the spinal cord. That's called the central process of the sensory axon. That central process will grow towards the developing spinal cord, will enter the developing spinal cord where the sensory gray meets the surface. It may synapse here. Some of them synapse there. Only some turn up with sending collaterals. That's all irrelevant for this course. It's all neuroanatomy. The important part is that the central process enters the dorsal gray and brings sensory information to the dorsal gray, or will. The peripheral process will have as its job bringing somatic sensation back from the muscles that are developing from these dermamyotome cells, or from the skin that overlies those muscles, or from the tendon or the fascia that overlies those muscles, or from the bones to which these muscles attach. So it's sending that out, and this one I've allowed to stop and say it is bringing back sensation from a muscle that will be derived from hypaxial dermamyotome. Here's another one which sends its peripheral process through the muscle to overlying skin. Some of them will send it to nearby bone. So there's a bunch of these somatic sensory axons coming back from the somatic structures of the body wall, some of which come from muscle itself, but some come from adjacent structures. And here is one that's bringing back sensory information from a muscle derived from an epaxial dermatometome. And here's one bringing back sensory information from the skin that overlies the muscle derived from that epaxial dermatometome. And there are thousands of these things. Okay? These are then, in blue, all of the somatic motor axons of this body segment, and in red, all of the somatic sensory axons of this body segment. Again, the spinal nerve is created to do the body wall. The body wall is overwhelmingly composed of somatic structures, and this is how those somatic structures get their motor innervation and give rise to sensory innervation, and how motor comes from the spinal cord and sensory gets to the spinal cord. This is a zone of crisscrossing between sensory and motor axons, because things that go up here to the epaxial region of the body wall, I will now define the epaxial region of the body wall as that portion derived from an epaxial dermatometome and everything in the immediate vicinity of those muscles that come from epaxial dermatometomes. And it's really the portion of the body wall which is dorsal to your vertebrae. The hypaxial region of the body wall are all the hypaxial muscles that came from the hypaxial dermatometome, plus all of the overlying skin and structures associated with those muscles. So the body wall is divided into an epaxial region, dorsal to your vertebral column, and then everything else, which is the hypaxial region, into which hypaxial dermatometome cells migrate. You'll note that every nerve going to the epaxial region of the body wall is both sensory and motor. Every nerve going to the hypaxial region of the body wall is both sensory and motor. But this thing is only sensory here, and this thing is only motor there, so they have to be a crisscrossing where to get these axons to the right place. Here is drawn in thousands of these somatic sensory axons, thousands of these somatic motor axons. Here's the zone of crisscrossing. Here is the branch that goes out to the epaxial body wall, bringing somatic motor to it and somatic sensory from it. Here's the much bigger branch going out to the hypaxial portion of the body wall, bringing somatic motor out to it and bringing back somatic sensory from it. This zone of crisscrossing is called the spinal nerve, and I put parentheses, sensu stricto. Now, sensu stricto is Latin for in the strictest sense. We have had people on examinations label this thing as sensu stricto. That's not the name of this thing. The name of that thing is the spinal nerve. All right? And I say sensu stricto because, you'll see in a second, this is very obviously the dorsal root of that spinal nerve. It is the thing on the dorsal side which contributes to it. This thing here is a ganglion embedded in the dorsal root of the spinal nerve. That's where the cell bodies of all these sensory axons lie. This thing is the ventral root of the spinal nerve. It's all somatic motor at this point in time. And it is the root of this thing called the spinal nerve. This thing here is a branch of this little tiny spinal nerve, which is going to the epaxial region of the body wall. It's called the dorsal ramus, which just means dorsal branch. That's what ramus means. It means branch. This is the dorsal branch of the spinal nerve for all of the epaxially derived muscles and the overlying skin and nearby bone and stuff. And this bigger one, because the hypaxial dermatometome migrates much further and is a much bigger portion of the body wall, is called the ventral ramus of the spinal nerve. The sensu stricto means that that's really how people should talk about it, but it's not ever going to be talked about that way. The spinal nerve, by most people, is the whole thing. Its roots, its rami, and this thing called the zone of crisscross, all together. When people say the spinal nerve, they tend to mean the whole thing. So you just have to be aware, though, that in fact, and you'll see this in dissection, the medical students today, the dental students Friday, This is really a very, very small zone of crisscross. I mean, it just exists so things can get to the right place. It doesn't exist for any reason other than to allow things to get to one or the other region of the body wall. It's maybe a millimeter long. You got these dorsal roots and ventral roots come, they crisscross, and then they immediately divide. So the zone of crisscross, the spinal nerve sensu stricto, is very, very short. Here's just a picture of what this will look like. I've shown now the hypaxial dermamyotome cells migrating all the way around the side and towards the front, because they do, and make up this whole portion of the body, which is the hypaxial portion of the body wall. And the epaxial dermamyotome cells just migrating dorsal to the vertebrae and the ribs and becoming the much smaller epaxial region of the body wall. And here is our dorsal root with its ganglion and our ventral root, and here's the zone of crisscross, and here's the small dorsal ramus going up to innervate epaxial musculature, and the small, much larger ventral ramus going to innervate all of the large expanse of hypaxial musculature and overlying skin and associated structures. And now I just put them in brown to show that all those dermamyotome cells become muscles. We tend to think of muscles as brown. So I put it in brown. Here is that picture showing every one of these spinal nerves. Every dermamyotome must have a spinal nerve associated with it. Every spinal nerve associated with it divides into a dorsal and ventral ramus for the corresponding epaxial and hypaxial regions, or whatever is the right way to say it. So I've shown all the dermamyotomes on the left and all the dermamyotomes on the right, and I've shown the spinal nerve having grown out and dividing into a dorsal and ventral ramus, the ventral for the hypaxial and the dorsal for the epaxial portion of those dermamyotomes. There are exactly the same number of spinal nerves as there are dermamyotomes. And that being the case, since there are 8 cervical dermamyotomes, there are 8 cervical spinal nerves. There are 12 thoracic dermamyotomes, there are 12 thoracic spinal nerves. There are 5 lumbar dermamyotomes, there are 5 lumbar spinal nerves. You must get annoyed saying this, you must get annoyed hearing this. There are 5 sacral dermamyotomes, there are 5 sacral spinal nerves, there's 1 coccygeal dermamyotome, there's 1 coccygeal spinal nerve. All right? The problem is, you have 8 cervical spinal nerves and only 7 cervical vertebrae. The first cervical spinal nerve comes out cranial to the first cervical vertebra. The second comes out cranial to the second cervical vertebra. The 7th comes out cranial to the 7th cervical vertebra. The 8th cervical spinal nerve comes out cranial to the first thoracic vertebra. And after that, all the spinal nerves come out caudal to the corresponding vertebra. And that's simply the convention by which these dermamyotomes were named. All right? And you can't change it. You have 8 cervical spinal nerves. Their relationship to the vertebra of the same name and number is different than the rest of the entire body. Think about it. Now I've just gotten rid of all the, because I want to show you what happened in the adult. I got rid of all the dermamyotomes. I said, let them, they've all migrated, I'm just concentrating right now. And I just made short ribs, I shouldn't have done. This is supposed to be an adult. Here are 7 cervical vertebrae here. Here are the rib-bearing vertebrae, the 12 thoracic rib-bearing vertebrae, 5 lumbar, 5 sacral, whatever. And here are the spinal nerves. I guess I should go back on. When this is all happening, these spinal nerves run a very straight course, very straight to the respective dermamyotome, which is very close to them. But what happens in the entire 9 months of development is that the vertebral column grows much, much greater in length than does the spinal cord. So as a result, the hole by which this nerve exits from the vertebral column no longer corresponds in position to the origin of that nerve from the spinal cord. And you see, here's a case of an adult where the vertebral column is much longer than the length of the spinal cord. So all of these nerves, which exited here in the beginning from a portion of the neural tube right near them, now actually sort of get stretched out as the vertebral column lengthens so much more than the spinal cord does. And so from the lower part of the spinal cord, and in an adult, the nerves have to descend, or do descend, to get out from the hole they originally went out when you were just an embryo and everything was the right length. All right? As a result of that, you find that you open up, you're going to do your laminectomies today, you're going to remove the laminae of the vertebrae, you're going to see a whole region from the L1, L2 disc down, which is just composed of nerve roots, dorsal roots and ventral roots, descending to leave from the hole that they left at before this differential growth occurred. That whole zone of descending ventral roots and dorsal roots is called the cauda equina, which means the tail of the horse because it looks like a horse's tail. Only exists because of the differential growth of the spinal cord and vertebral column in fetal life. There's a bit of pia which comes down the middle, which I didn't label here, called the film terminale, but I'll get there. Don't have to worry about that now. As a result, you know, when you were a young embryo, the first sacral segment of your spinal cord was right here, right, where this first sacral, right, just caudal to the first sacral vertebra. All right, that's first sacral vertebra. You know, the first lumbar region of your spinal cord when you were a little embryo was just caudal to the first lumbar was just caudal to the first, say it in English. The first lumbar segment of the spinal cord, which is the segment that gives rise to the first lumbar nerve, was immediately caudal to the first lumbar vertebra. As a result of this differential growth, the spinal cord segments, which give rise to the corresponding nerves, are no longer in close relationship to the same vertebra number. And this is the rule of ones, which is what I use to remember where things are. In an adult, as a result of the differential growth, the top of the first cervical segment of the spinal cord, which gives rise to the first cervical spinal nerve, is more or less equivalent with the top of the first cervical vertebra, or the base of the skull. The top of the first thoracic spinal cord segment, which gives rise to the first thoracic spinal nerve, is coincident with the top of the first thoracic vertebra. So there's been very little shortening in the thoracic region, in cervical region, sorry. Very little shortening in the cervical region. Most of the shortening occurs near the back. And as a result, the top of the first lumbar segment corresponds to the top of the 11th thoracic vertebra. Someone shoots you through your 11th thoracic vertebra, they will in fact damage the first lumbar segment of your spinal cord, not your 11th thoracic segment. And the top of the first sacral segment of the spinal cord, which gives rise to the first sacral spinal nerve, is equivalent to the top of the first lumbar vertebra. The rule of ones is it's 1, 1, 1, 1, 1, double 1, 1, 1. All right. First segment, C1, T1, L1, S1, the corresponding vertebrae would all have ones in their name. You just have to memorize which ones. Only ones. Here are the membranes which envelop the spinal cord. There's our spinal cord. The spinal cord itself has a tough, fibrous outer sheath called the pia. You cannot remove the pia from the spinal cord without destroying the spinal cord. Then there's some fluid. It can be cerebrospinal fluid, might be in your case embalming fluid, but there's some fluid. And the next layer of membrane is called the arachnoid. Very, very delicate, like a soap film almost.
I am about to state that on the outer surface of the arachnoid is the dura. In truth, between the arachnoid and dura is a thin layer of flat cells that comprise the dural border cell layer. This layer is not macroscopically visible and is very easily split, giving the false impression that only a film of fluid separates arachnoid and dura.
And then on the outer surface of the arachnoid, you will see, this is the first thing you will see, is this very tough structure called the dura. You will cut through the dura delicately, hoping to see the arachnoid, which adheres by fluid tension only adheres by fluid tension to the deep surface of the dura. And then once you pull the arachnoid back, if you haven't destroyed it because it's so delicate, you will see, if you're at the region of the spinal cord with the pia on it. These nerves, dorsal roots and ventral roots, pierce or push out in front of them dura and arachnoid. And then you will see then, coming out of this dural sac, a sleeve of dura which envelops dorsal roots and ventral roots. The dorsal roots and ventral roots don't actually fuse to make the spinal nerve sensu stricto until the distal edge of the ganglion. So you will see a sleeve of dura. You'll know that within that sleeve is a separate dorsal root and ventral root. Then you will see a bulge in that sleeve of dura. And you will know that that bulge signifies the existence of the dorsal root ganglion. But still, when you see that bulge, you'll know the dorsal root and the ventral root are actually separate. And they're separate by a connective tissue septum. And only at the distal edge of that bulge do you have the zone of crisscross. And that's when you will see the spinal nerve sensu stricto. And immediately you see that zone of crisscross, you'll see a dorsal ramus and a ventral ramus branch off. This is less than a millimeter, a millimeter long. This just reminds you, if you took a section through that dural sleeve that comes out, it actually would have a separate dorsal and ventral root. If you took a section through the lump, it'll be a separate dorsal root ganglion and ventral root. Still no zone of crisscross. And then beyond that, you have the spinal. That's what you'll see in lab today.